Hello everyone. Today we're going over fungal life cycles, primarily Ascomycetes and Basidiomycetes. So to start off, we're going to start off with a myospore. A myospore is a spore that's produced by meiosis, or by sexual reproduction. We're going to go over that in a second. But we have two myospores here and one here. Let's say they germinate nearby each other. When they germinate, they put out a germ tube, which turns into a hypha. And now we have hyphae comprising mycelia that begin to spread out and eventually meet. When they first germinate, they are both going to be homokaryotic. They're also going to be monokaryotic, meaning we're going to have all the same nuclei within each cell of each hypha. And within each separate hypha, which in each haplotype, right, each two genetically unique spores, two genetically unique fungi, within each of these, each cell is going to be limited to one N. Each cell is going to have one nucleus, and each nucleus is going to be haploid, meaning it has one set of chromosomes, not two. So these two hypha germinate, and eventually they meet up. And so we're going to assume they're vegetatively compatible. We're going to assume that they can actually conjoin. They can go through anastomosis and plasmogamy. And when that occurs, it occurs simultaneously. So anastomosis refers to when two linear hypha, right, they meet each other. We can see a depiction of that down here in a few locations, right? Here are hyphae of two different fungi, or perhaps even the same fungus, meeting each other and going through anastomosis, which is where two fungi, their hypha meet, and they conjoin essentially. They mix cellular cytosol, essentially, and they become one hypha. And the mixing of cytosol is plasmogamy. The joining of those hyphae is anastomosis. So these two processes can never be separated. They happen simultaneously, anastomosis and plasmogamy. When these two hypha meet, they go from each being haploid to sharing a hypha now, and each of these cells has two haploid nuclei, which means each cell is dikaryotic, and the hypha altogether is a dikaryon, which is N plus N, right? And this is different from diploid. This is not 2N. We do not have a single nucleus with two sets of genetic two different sets of chromosomes. Instead, we have a single cell. Each single cell has two nuclei. Each nucleus is haploid. And this dikaryotic phase is unique among fungi to the Ascomycetes and the Basidiomycetes. This is actually a synapomorphy, right? This joins this monophyletic clade together. So dikarya is the clade that is comprised by Ascomycetes and Basidiomycetes, or Ascomycota and Basidiomycota. So, Ascomycetes and Basidiomycetes are both capable of going through anastomosis and plasmogamy and forming a dikaryotic cell and a dikaryotic hypha. This is important because this is the step that occurs before karyogamy, which you'll see down here. We're going to go into this in a second. But I want you to recognize that a dikaryon is the nuclear state where you have two haploid nuclei within each cell. This is an important characteristic, and you will get points off if you don't know this. So this is just a kind of abstract depiction of a fungus life cycle, a Basidiomycete or an Ascomycete. We have two haploid cells. We could call these spores. These could also be representations of two hypha. They are 1N. They are haploid. They go through plasmogamy. When plasmogamy occurs, like we just talked about, anastomosis also occurs simultaneously, and now you end up with each cell having two different haploid nuclei. So each cell has two nuclei, and Ascomycetes and Basidiomycetes, they do have pores in their septa. They are functionally cetocytic, so this dikaryotic stage is maintained by the fungus. Even though nuclei could flow through the hypha if they wanted to, they don't. Or they do minimally. And the fungus typically maintains this heterokaryotic, two different haploid nuclei within each cell on a dikaryotic hypha. So we have a dikaryotic cell, and then we have karyogamy. And karyogamy often, this will occur in the hymenium of an ascocarp or an ascoma or a basidiocarp or a basidioma. So we'll look at this big picture. But basically, karyogamy is similar to fertilization in that it results in a diploid nucleus. However, it's different from fertilization in that it doesn't start from two separate haploid cells. It starts from cells that are dikaryotic, that each cell has two haploid nuclei. And this is a very important distinction between fertilization and karyogamy. So karyogamy occurs. You end up with diploid cells. Diploid cells are 2n, right? We go from n to n plus n is what this should say. I think it should. It doesn't say it next to my head, but it should. You go from n to n plus n in this dikaryotic. Oh, here it is, n plus n. And then it goes to 2n, which is the diploid cell. Immediately, practically, after karyogamy occurs, meiosis occurs. And meiosis occurs to produce myospores. In Basidiomycetes, meiosis occurs, and then spores migrate, or nuclei migrate into spores. You have 4 spores because meiosis results in 4 cells. In Ascomycetes, right, meiosis occurs, producing 4 cells, producing 4 spores. Meiosis occurs, producing 4 nuclei, and then mitosis occurs, doubling those nuclei. So now you have 8 nuclei, and those migrate into spores that are released from the asci. Sorry about the confusion. Meiosis occurs, producing these haploid cells, the myospores, which are released via ascospores and basidiospores. So here we are not depicting conidia. We're not depicting asexual spores at any point in this life cycle. This is primarily depicting the sexual life cycle. And there's some, vegetative part of the life cycle here, but the sexual life cycle of an Ascomycete or a Basidiomycete. So here we have a Basidiomycete life cycle. Here are those 4 basidiospores that are produced as a result of meiosis, of meiotic division. These basidiospores are dispersed through air, wind, water, and they eventually germinate. They produce these hyphae. These hyphae will meet, and they will go through plasmogamy and anastomosis. They will join, and they'll go from being each n to together n plus n. So now we have a hypha that is dikaryotic, n plus n. And often Basidiomycetes will actually live for a while in this dikaryotic phase, which is different from Ascomycetes. Ascomycetes typically like to quickly produce an ascocarp right after going through plasmogamy and anastomosis. Basidiomycetes like to subsist as a dikaryotic hypha for longer. This is a distinction. So a young Basidioma, synonymous with basidiocarp, is produced, and this basidiocarp has a hymenium, a fertile layer. This fertile layer is pretty much where karyogamy and meiosis occurs to produce your myospores. So this basidium is located in this hymenium, in this fertile layer. So I want everyone to understand this, that this whole part of the life cycle occurs only in this hymenium, only in the fertile layer of your basidioma or your basidiocarp. This is occurring primarily in just the vegetative hyphae of 2 growing fungi, of the mycelium of 2 growing fungi. Hopefully this makes sense. Within this fertile layer, karyogamy occurs, right? This is when you go from n plus n to 2n. This is akin to fertilization, but different, as we just talked about. Now you have a single diploid nucleus within this cell. Now you have meiosis occurring, right? Meiosis occurs in 2 parts, turning one nucleus, one 2n nucleus, into 4 haploid nuclei. These 4 haploid nuclei migrate through the sterigmata, end up in these basidiospores, and the basidiospores are released to begin the next life cycle. Okay, now we're looking at an Ascomycete life cycle. And a lot of things are similar, but there are a few distinctions I want to outline. We're not going to talk too much about the trichogyne or the antheridium. These are things you can learn in mycology if you want to. What I do want you to know, for identification purposes largely, is we have our ascus, so akin to a basidium, right? We have a basidium here, we have an ascus here. An Ascomycete's ascus produces 8 myospores compared to the 4 myospores in a basidium. Those myospores are called basidiospores in Basidiomycetes, and they are called ascospores in Ascomycetes. And so Ascomycetes produce 8 per ascus, as opposed to Basidiomycetes, which produce 4 per basidium. This is just a useful delineation. The next thing I want to talk about is the presence of conidia. So conidia, which are produced on conidiophores, which are conidia-bearing structures. Conidia are asexual reproductive spores. These are mitospores, mito with a T. They are produced only by meiosis. Meiosis does not occur to produce conidia. And conidia are primarily produced by Ascomycetes, rarely produced by Basidiomycetes. And they are often an important part of the life cycle of a lot of the pathogens we're studying. Most of the pathogens we study, with the exception of rusts and agarics, mushrooms, and etc., most of the pathogens we study are Ascomycetes, and so asexual spores, conidia, mitospores, are an important part of most of their life cycles, or many of their life cycles. And they can be useful for identification as well. So the same thing essentially occurs. Plasmogamy and anastomosis occurs. We do have this little asexual part in between, but otherwise the sexual life cycle is not any different. We have these specialized structures we're not going to go into, take mycology. Anastomosis and plasmogamy occur. As we talked about before, Ascomycetes like to go right from dikaryon. They like to immediately go through karyogamy. They don't like waiting around vegetatively in this dikaryotic stage, in this N plus N stage. They would rather quickly form an ascoma or an ascocarp, and they would rather quickly go through karyogamy, meiosis, right? Meiosis 1, meiosis 2, and then, unlike Basidiomycetes, they go through mitosis, right? So we have these four nuclei in this cell, and then it doubles those 4 to 8, and then we have 8 ascospores within this ascus. So hopefully you can glean some of these details, right? In Ascomycetes, conidia are much more common. In Ascomycetes, they don't like to linger around in this dikaryotic stage. In Ascomycetes, we have 8 versus the 4 myospores that are produced from each ascus, or analogously Basidium. Cool. And here's a good overview, right? I want you all to know what phyla, and here we have Oomycetes. These are not fungus. These are not fungi, of course, but we still have myospores. We have spores that are produced through meiosis sexually, and we still have mitospores in Oomycetes. And Oomycetes are very similar to fungi in a lot of ways, but of course they are analogously similar to fungi. They are an example of divergent evolution. They do not gain these shared attributes from a common ancestor. They develop them separately, convergently. Convergent evolution. They are not an example of divergent evolution. They're an example of convergent evolution. My mistake. Okay, and we've just been over this, so look through this. I want everyone to be able to draw through an Ascomycete or a Basidiomycete life cycle, and be able to label everything from plasmogamy to anastomosis to karyogamy, meiosis and mitosis if mitosis occurs. So if it's an Ascomycete, you're going to be putting mitosis here as well as meiosis. And if it's an Ascomycete, you have to know these asexual spores, these conidia, are produced, and they are mitospores. So I want you to be able to draw through that. What else do we have, right? These are all pretty good. Okay, so now we're going to talk about vaguely. So now we're going to talk about the life cycle of an Oomycete. Oomycetes, just the similar way that Ascomycetes and Basidiomycetes started meiotically by ascospores and basidiospores, respectively. You can think of Oomycetes starting with these oospores. And these oospores are the result of meiosis and then fertilization. And they are dispersed. They are sexual spores that are dispersed. Unlike in Ascomycetes and Basidiomycetes, these are not haploid. These are diploid. And so you'll notice that this organism, this protist, this tremenopile, Oomycetes, spends the majority of its life as diploid. It only ventures into being haploid for a moment here when it's reproducing sexually, but then it spends the majority of its life as diploid, just like us, right? You know, we only spend a small portion of our time as haploid. We do this as sperm and egg, right? So oospores are released. They will grow hyphae and mycelia. They can produce chlamydospores, which are heavily reinforced. Often there's a hardened exterior. Often they're melanized. These are long-term storage. These are dormancy structures. So chlamydospores are a type of asexual propagule, mitotically produced, and they will also produce sporangia. Sporangia house zoospores. Sporangia are also sometimes called zoosporangia. They house zoospores, which are these flagellated, motile, asexually produced spores that are distinct in Oomycetes. So we don't have flagellated spores in fungi. We do in chytrids, right, in Basidiomycete. We do in basal fungi, but flagella are lost relatively early in fungal phylogeny. So this is a pretty big distinction. We have motile flagellated zoospores that essentially will swim through water films. This is why they rely heavily on excess moisture. They'll swim through water films in the soil, and eventually, as in the case with some of the root pathogens we've studied so far, right, Phytophthora cinnamomi, they will form a cyst on the roots of their host. They will grow hyphae. They will form an oogonium and an antheridium that will essentially join, and they will be the site of meiosis and fertilization. And here we have fertilization, where this oogonium, which is like a womb of sorts, ends up being filled with oospores. And oospores are the process of first meiosis between the antheridium and the oogonium, then fertilization between the propagules of each. And now you have this oogonium mature and filled with oospores, which are the meiotic spore that now can disperse into the wild and start this over again. So on one of these slides, here's up close, on one of these slides, we have all this stuff kind of talked about. But the important things I want you to glean from this, right, is we do have these asexual zoospores, which are motile, and they have flagella. We have the majority of the life cycle of an Oomycete is spent at a diploid state, as opposed to an Ascomycete, which spends the majority of its life, let's say, in a haploid state, and a Basidiomycete, which spends maybe the majority of its life in a dikaryotic state. What else do we have? We have fertilization actually occurs, right? In Ascomycetes and Basidiomycetes, fertilization doesn't occur, karyogamy occurs. Because karyogamy moves from n plus n to 2n, fertilization moves from n and n separately, not n plus n, but two haploid nuclei in two separate cells, not in the same cell, in two separate cells. Fertilization occurs when you have those two cells coming together, and there's no intermediate state where two nuclei occupy one cell. Immediately, you go through fertilization and you go from n to 2n. And this is a big distinction from Ascomycetes and Basidiomycetes, from that dikarya clade, which go through, they have an intermediate stage that in Basidiomycetes is quite long, and Ascomycetes is relatively short. They have an intermediate stage where two haploid nuclei occupy each cell. This is the dikaryotic stage of a hypha. So I want to make that distinction clear as well. And you could think of zoospores as analogous to conidia and Ascomycetes. You can think of oospores as analogous to Basidiospores and Ascospores in Basidiomycete and Ascomycete. I'm losing my voice. Okay.